Next, Alison Harrill is a geneticist at the NIHS, and she'll be speaking to us about defining precise in environmental toxicology and public health using diversity outbred mice to mimic human susceptibility to environmental chemicals.
Great, thank you for the introduction, and thank you to the organizers for having me here. So I'm gonna flip the switch a little bit and move away from medicine to talking about chemical risk assessment for a minute, and I'll give you a little bit of background. First, my standard disclaimer. Moving on from that, so human health chemical risk assessment is a field in which we very rarely have human data. We'll, in some cases, most cases, never have human data. And what we're thinking about are those many thousands of chemicals in commerce. So these are things like flame retardants, pesticides, dyes, manufacturing catalysts and other materials, and so on, or naturally occurring substances in the environment like arsenic, lead, diesel exhaust, which is not natural but something that people get exposed to. And we're also thinking about multiple scales and routes of exposure. So are we talking about occupational exposures, which are long duration, high dose, where you have to think about risk management techniques? Are these the individual users, like me going home, spraying some pesticide around my house, maybe once a month, which would be a short-term exposure, or is this a community exposure? And then as far as routes, is it oral, is it inhaled, is it dermal, for example, and does that change based on the life stage of the individual who's being exposed? For instance, infants who are crawling on the ground, putting a lot of things into their mouths. And so there's lots of data that we collect to inform risk assessment for individual chemicals. Here we're thinking about the hazards, so what are the target tissues, what are the health outcomes that arise upon exposure. We're thinking about dose and potency, so at which exposure, whether that's cumulative or short-term, is there an undue risk? We're thinking about susceptible subpopulations, so we're thinking about age dynamics and differences in susceptibility for the very young or the very old, for example, or life stage. Is there maternal exposure effects on the developing fetus, for example? So there's all of these elements that come into a role when we think about how to regulate and inform risk assessment for chemicals. And all of these data that inform these processes typically come from animal guideline studies, similar to what's done in the pharmaceutical safety arena, and increasingly in vitro studies are coming into play as well. So we've had a lot of discussion over the last 2 days about gene-environment interactions, and these are important as well for chemical safety assessments. So in 2009, there was an NRC report that advised that attention should be directed to vulnerable individuals and subpopulations that might be particularly susceptible. And some of that susceptibility may be based on the individual's genetic background. Understanding influence of genes can help us to inform uncertainty estimates. So for example, it's been shown in prior studies that exposure to methylmercury there can be a 50,000-fold variation in susceptibility across populations. And in exposed populations, about 5% of individuals are on average 25-fold more susceptible than other individuals in the population. And so if we can come up with avenues to understand this population dynamics in response, we can actually use that information to inform where we ought to be setting safe exposure thresholds for a lot of these chemicals. So diversity is rarely incorporated into standard safety assessments. At the National Toxicology Program, we typically use for mice the B6C3F1 hybrid mouse, which is a F1 cross between black 6 and C3H. So across all of these individuals, they're all genetically identical. Our rat model is the Sprague-Dawley rat. The Sprague-Dawley rat has been well characterized genetically and phenotypically, and so we know that there are limits to the amount of genetic diversity that's present, in part due to selective pressure that the population has undergone. And so there's a low amount of allelic variation, and there are many regions within the genome that are identical by descent. There's not a lot of, or essentially no genetic variation within these long regions that you can interrogate. And then in the case of human cells, when these fit-for-purpose assays that are being developed, oftentimes we're looking at data from a single donor or maybe a few donors. And in the past, this has been considered a feature, not a bug. So we wanted to constrain variability within these populations so that we can have greater confidence in understanding background variation, as Dr. Berridge had alluded to in his talk. But new tools have made it a more tractable avenue to explore to actually start looking at genetic susceptibility. So one of these avenues is diversity outbred mice. These were developed by the Complex Trait Consortium, which had multiple stakeholders, including the Jackson Laboratory, University of North Carolina, and others. And so what they had hoped to do and what they achieved was to develop a rationally designed population that mimics human genetic diversity, but in a much more randomized way. So the old way of looking at population diversity in mice was really based on using multiple inbred strains, pulling strains off the shelf like C57 Black 6, 129, and others. But in this new population, what was done was there was an 8-way cross of 8 genetic co-founder strains. These co-founder strains incorporate genetic material from 3 different subspecies of Mus musculus, including Musculus domesticus and Castaneus subspecies. So you get a lot of diversity by intercrossing these 8 genetic founder strains. And so this resulted in 2 sister populations, one of which is the collaborative cross, which is comprised of over 100 recombinant inbred lines, which encompass a reshuffling of genetic material from the 8 founder strains. And the other is the diversity outbred mice, which I'll focus on in my talk. So this is a very large outbred population in which each individual is genetically unique, but across individuals you have a very large genetic diversity. And one benefit of this population as well is that the proportion of rare alleles is on average about 12.5%. Compare that to humans where the frequency is typically less than 5%. And so you can look at the querying a genetic space with far fewer individuals in the mice than you would need in a comparable human study. So I mentioned that the diversity outbred was created by intermixing or interbreeding 8 genetic co-founder strains. Here's the 8 strains. I won't walk through this, but as we know, each strain has a propensity to certain diseases. Maybe they're age-related, maybe they're chemically or diet-induced. And so what this enables us to do is look at chemical toxicity on a background of comorbidities that is very similar to that of the human population. We've thought quite a bit about how we would use mouse populations in conventional risk assessment paradigms. And so what I list here are 4 major buckets that we try to collect data for when doing a chemical risk assessment. So for exposure assessment, if we're using a mouse population, we can look at population-based differences in pharmacokinetics and dynamics. We can also use a population to establish either biomarkers of exposure or biomarkers of effect that may have some more precise measurements than human populations, and I'll show you some data to that effect later on in the talk. We could potentially use these mice to identify chemical hazards that our conventional models miss. And I won't show those data today, but in my lab we've looked at drugs that have failed during clinical trial phases, put them in diversity outbred mice, and shown that we could have gotten the relevant clinical effect that was missed by conventional preclinical screening. We can look at dose responsiveness and potency and how that changes in a population space if you're including genetically sensitive individuals in the population. And then we can look at mode of action. So if you can query a population, identify who's susceptible, you could perhaps use Jim Stevens' Gymgenuity analysis and look at pathways that are distinguishing the susceptible versus the non-susceptible individuals. So there's a variety of avenues that we can explore here. So I'll give you some usage cases for how, in my laboratory, we've used diversity outbred mice. So the first case, it sort of dovetails with our discussion yesterday. So how can we use mouse populations to identify specific gene variants that influence susceptibility to drug and chemical toxicity. So one example we've published a couple years ago was actually looking at herbal supplements containing green tea extract. We were interested in this because we were working with a clinical consortium, the Drug-Induced Liver Injury Network, who were interested in coming up with genetic tests that could potentially inform causality assessments. So when a consumer comes in, they're taking a variety of herbal supplements and perhaps pharmaceutical drugs as well, and so they would want to establish a causal link to the green tea extract containing herbal supplement. So when we actually applied a green tea extract component, epigallocatechin gallate, to diversity outbred mice, gave them the same dose over a short-term duration of oral exposure, what we saw in this population is about a third of the animals were completely resistant to the toxicity if you look at percent of liver necrosis, and about 2/3 were susceptible, but there's a break point here. We have a very sensitive, small subpopulation of individuals which were analogous to those very severe idiosyncratic liver cases that arose where consumers were presenting at the hospital with this. And so what we can then do is genetic analysis using, first we genotype the animals using the mouse universal genotyping array, which is a specialized array for the system. And then we could do genome-wide association analysis. Here we found a peak on chromosome 4. When we actually looked at that interval, what we found is that here we have the founder effect plots, and the various colors indicate the relative effect of having inherited that chunk of DNA from each of the 8 founder strains. So does it make you have a higher degree of liver necrosis or a lower degree? And so what you can see here at our QTL interval, if you had genetic material donated from the NOD founder, you were less susceptible to the toxicity induced by green tea extract. And then we could, within that space, identify those genes in that region and figure out which ones have an NOD private SNP. We then went into a clinical population to validate some of our candidate SNPs from that region. So not a genome-wide approach, but a very targeted approach where we first started with the mice and then moved into humans. And we found associations with 3 genes, one of which is involved in circadian rhythm signaling, which is period 3, and mitofusin 2, which is involved in mitochondrial autophagy, essentially. And then we did subsequent studies where we actually interrogated the capacity of green tea extract to cause deficiencies in mitochondrial bioenergetics, and so we were able to come up with a causal link to mitochondrial toxicity in the susceptible patients. These are some data that were not from my lab. This was generated by the Jackson Laboratory, headed by Dan Gaddy and Gary Churchill. And so they have also begun investigating using diversity outbred mice to understand susceptibility to various toxicities. Here, this is a study that was recently published where they looked at chemotherapeutic-induced neutropenia. And what I just wanted to quickly show here is if you look at those gene regions, this is the chromosome number, or the numbers down here. If you look at the gene regions that are associated with neutropenia for each of these drugs, it actually differs between drugs. So that indicates that there's a different mode of action. And by using this population approach, not knowing who's susceptible up front, we can identify these mechanistic links here. Okay, so the next usage case I wanted to talk about is using a population like this to evaluate novel toxicity biomarkers. So many groups are looking at identifying new and improved biomarkers for various organ systems. In my laboratory, we've done a lot of work on liver and kidney biomarkers, but a lot of these investigations are being conducted in limited genetic contexts, like Sprague-Dawley rats, and there are major efforts now to translate those biomarkers from rodent studies to the clinic. And so I thought, well, why not use this population approach to actually look at potential clinical relevance of some of the biomarkers that people have become excited about. So the biomarkers we looked at in the study that I'm going to highlight, again, are renal biomarkers. So we compared against gold standard markers like creatinine and blood urea nitrogen, which are typically used in preclinical tox studies for rodents. And then we compared those against urinary protein biomarkers that were recently qualified for use in preclinical tox studies in the rats. And so those are all listed here. These have, in various ways, in at least rat models, been shown to be more sensitive and specific than BUN and creatinine, and they also, in some cases, inform the site of injury to the renal nephron. In addition, we looked at microRNAs, which have been looked at in a major way as emerging biomarkers, both in the serum and the urine. In this case, we looked at the urine. So in this study, we used cisplatin as a prototypical toxic agent. We looked at proximal tubule injury primarily. That's what we were hoping to elicit. We had a pretty large N number because these are outbred mice. Your variance is high, and so we wanted to adequately power the study. And we also, we treated the animals on day 0 and then took urine before necropsy on day 3. So here's what the conventional biomarker looks like. This is BUN. And you can see in this population that there's a variability in susceptibility to cisplatin-induced kidney injury here as defined by BUN increases. And there is a subpopulation of individuals that had an increase in BUN. If you look at the histopathology, what we primarily saw was renal tubular degeneration and necrosis. But the severity depended on the individual animals. So in about 26 individuals in the cisplatin-treated group, we had no pathology, so they were resistant to the toxicity. In 17, there was a minimal change, and in 9, there was a mild or a slightly more severe necrosis that we observed. And so we actually looked at binning the biomarker values benchmarked to the severity of the underlying necrosis of the renal tubular cells. And so if you look at BUN, just as we expect and is argued by many of the individuals, including myself, who have looked at developing new biomarkers, BUN did not increase until we had grade 2 necrosis. So that was expected. What wasn't expected was that we saw the same trend for all of the urinary biomarkers that we looked at. So there was elevation in several of these that are associated with proximal tubular injury, like renin and beta-2 microglobulin, for example, but there was no elevation at the lower levels of necrosis. And so the enthusiasm for translating these to the clinic might perhaps be less, given that the performance was really not that much better than BUN. The same was true for urinary microRNAs that we looked at. So here, in this case, we're looking at CT values because this is an RT-PCR assay. So less is more in this case. And again, we didn't see elevations in the urine until a grade 2 necrosis. Interestingly enough, if you look at the ROT curves, which is commonly done to evaluate biomarker performance, these aren't bad. We get RX values between 0.7 and 0.82. P-values are significant, but yet when you actually parse out the underlying histopathology, we really don't see as much promise in some of these. Now, the caveat, this is mice. The clinical experience might be somewhat different, and so I think there's more work that needs to be done. But I think this really shows how we could potentially use this type of model to evaluate biomarkers of various stripes. So in the last couple minutes that I have, I'll just touch on an avenue that we're starting to move into at NTP. We don't have a lot of data here yet, but I think this is a very promising usage case. So I've talked to you a lot about how we've used in vivo data in various contexts. We're also thinking about how we can bridge to the in vitro space and use diversity outbred cells for medium throughput types of investigations. And so largely what we're focused on is understanding where the point of departure is. If I can go back. Right. So where's the point of departure? At what dose do we start to see an adverse effect that is biologically relevant? And we can define where the biological relevance is. But if you start looking at a population space, the variance in any of these points is going to be much greater than in our conventional models. And this was actually used successfully by Jeff French and colleagues who are at NTP. So this was an actual in vivo study, but it illustrates this point. So in this study, they were looking at benzene toxicity, and they did a dose-response analysis looking at numbers of mononucleated reticulocytes following inhaled exposure to benzene. And then benchmark dose modeling was done here to look at points of departure for chemical potency. And the interesting thing is, when you look at the human data, the benchmark concentration for where you start to see an effect in the small numbers of individuals that we were able to interrogate is 7.2 parts per million. But if you look at the calculated value using diversity outbred mice, which expands your space, it's orders of magnitude lower. And so can we use this type of paradigm to inform risk assessment for humans so that we can perhaps be somewhat more protective than we have been previously? So as I mentioned, we're looking at in vitro culture systems. I'm just going to breeze through this because I'm over time. So we're starting to look at screens for developmental neurotoxicity, incorporating genetic sensitivity into the model using diversity outbred cells. So we have neural progenitor cells that were derived from embryonic stem cells collected from these mice. They're highly genetically characterized. And we have done some initial scoping studies with several prototypical neurotoxic compounds to look at heritability of response. And we're actually starting to move into phase 2, where we're collaborating with folks at the FDA and the EPA together with us here at NIH to identify a paradigm by which we can begin developing these in vitro population screens, and how do we analyze and interpret the data in the context of human health risk assessment. So with that, I'll acknowledge several collaborators and trainees in my laboratory, and I thank you for all your attention.